Welcome to today's Explainer. We're setting out on a truly fascinating journey. Whether it's tracking down a banned substance in an athlete's blood, hunting for a microscopic pollutant in our drinking water, or figuring out the exact molecular structure of a brand new life-saving drug, scientists face one absolutely massive challenge. They have to see the unseeable. And to identify these compounds that are completely invisible to the naked eye, they rely on literally one of the most powerful analytical tools ever invented.
Now, I know what you might be thinking. When I hear the word mass, my brain usually jumps straight to a quiet Sunday morning. But no, we definitely aren't talking about a church mass today. We are talking about the fundamental physical weight of atoms and molecules.
So let's course correct to the actual scientific definition here. Mass spectrometry is an analytical technique that measures the mass-to-charge ratio of ions. By ionizing chemical species, basically giving them an electrical charge, and sorting them based on this ratio, we can figure out their exact molecular weight and structure. Honestly, think of it like a highly sophisticated microscopic scale that tells us exactly what tiny pieces make up the whole puzzle. Here's the roadmap for our sample's journey today. First, unlocking the invisible. Second, the core principles. Third, inside the mass spectrometer. Fourth, making sense of data. And finally, real-world impact in the future. Let's get into it.
Section 1. Unlocking the invisible. The challenge of analyzing chemical compounds. Okay, let's dive into this. The initial hurdle we have to jump is just that molecules are way too small to observe directly. We need incredible sensitivity to see their structures. But because the chemical world is so insanely diverse, there really isn't a one-size-fits-all starting point. Depending on the sample, we have to rely on specialized prep techniques before the mass spectrometry even starts. For instance, if we're dealing with volatile compounds, things that easily turn into gas, we use gas chromatography, mass spectrometry, or GCMS. If we have non-volatile or super-fragile molecules, we turn to liquid chromatography, or LCMS. And when we need absolute, pinpoint specificity, we use tandem mass spectrometry, or MSMS, which runs the sample through multiple stages of analysis. It's really all about getting the sample prepped perfectly for the journey ahead.
Section 2. The core principles. How exactly do we measure what we can't see?
To measure something this incredibly small, we have to rely on some basic physics. But there's a catch. Magnetic and electric fields, they absolutely cannot manipulate neutral molecules. They just don't care about them. So we have to transform these molecules into charged particles, or ions first. Once our sample is ionized, it shoots through a curved tube surrounded by a massive, powerful magnet. And because the ions now have an electrical charge, the magnet actually bends their path. It's brilliantly simple when you think about it. A magnet will deflect a lighter ion much more aggressively than a heavier one. Notice how the lighter path bends super sharply, while the heavier path barely curves at all. By measuring exactly where they land at the end, we can calculate their exact mass.
But hold on. How do we actually get that electrical charge onto the molecule in the first place? Well, we use different methods to zap our samples into ions. If we're working with relatively small, sturdy molecules, we typically use electron ionization, or EI, which basically just bombards the sample with a harsh beam of electrons. But if we're dealing with massive, delicate things like biological proteins, actually, scratch that. A harsh beam would literally shatter them to pieces. So we contrast that with electrospray ionization, or ESI. It's a much gentler technique that is perfectly suited for keeping those larger biomolecules intact.
Section 3. Inside the mass spectrometer. Following the physical path.
All right, now that we've got the physics down of making ions and bending their paths, let's follow our molecule deep inside the actual hardware. Every single mass spectrometer basically walks a molecule through 5 sequential steps. First up is the sample introduction system, where the material is injected. Second is the ion source, where those neutral molecules get zapped into charged ions. Third is the mass analyzer. This is the heart of the machine, where the crucial separation by mass-to-charge ratio happens. Fourth is the detector, which catches the sorted ions. And finally, step 5 is the data system, which is just a computer translating all this physical sorting into digital data we can actually read.
Now, what's really interesting about this next part is how we actually pull off step 3, the mass analyzer. Modern machines often use something called a quadrupole analyzer. Instead of a giant curved magnet, it uses 4 parallel metal rods. By rapidly changing the electrical voltages on these rods, it creates a very specific electromagnetic field. It's kind of like an exclusive VIP list. Only the ions with the exact mass-to-charge ratio we're looking for, the resonant ones, can successfully spiral all the way through to the detector. The non-resonant ions, they're filtered out and literally just crash into the rods.
To ground all this abstract theory in reality, Here's what this invisible journey actually looks like from the outside. These are real, highly complex mass spectrometry setups you'd find in a high-end modern lab today. Behind those sleek exterior panels are all the intricate vacuum pumps, ion sources, quadrupole rods, and ultra-delicate detectors that make this whole high-tech sorting process possible.
Section 4. Making sense of data. Reading the chemical fingerprints.
So once the molecule completes its crazy journey through the hardware, we finally move to the output. When those selected ions strike the detector, they generate these tiny, tiny electrical signals. The computer captures those signals and translates them into a mass spectrum. It's essentially a chemical fingerprint. Take a look at this simplified reading for pentane, for example. The graph charts the mass-to-charge ratio, the MZ, along the bottom axis, against the relative abundance on the vertical axis. Basically, every vertical line you see represents a literal piece of the molecule that struck the detector.
To decode this data, scientists are hunting for 3 critical clues. First up is the molecular ion peak, or M+. This represents the heaviest fragment, which is usually just the intact molecule itself. This gives us our total molecular weight. Second is the base peak. This one is super simple. It's just the tallest, most intense line on the entire graph, representing the most stable ion that was formed. And third, we have the fragment peaks. Remember, when a molecule is ionized, it often shatters into smaller pieces. These fragment peaks act exactly like puzzle pieces, revealing exactly how the original molecular structure was put together in the first place.
Section 5. Real-world impact and future. Why does this all matter?
So, what does this all mean? Why go through all this immense technical effort just to weigh some invisible molecules? Well, the crucial point is that being able to accurately analyze incredibly small samples has massive, literal life-saving applications. In clinical diagnostics, doctors are using mass spectrometer to analyze blood or urine, identifying specific disease biomarkers long before you'd ever feel a symptom. In environmental monitoring, it's the absolute gold standard for detecting dangerous trace pollutants hidden in our air and drinking water. And in pharmaceutical development, it's essential. It's how we assess the purity of new drugs and track exactly how our bodies metabolize them.
Of course, it's not all magic. This immense analytical power comes with some real trade-offs. On the plus side, it's a real game changer because it offers incredibly high sensitivity, dead accurate molecular weight determination, and you only need a minuscule sample to work with. But we have to balance that against the challenges. These instruments are staggeringly expensive. Plus, there is a huge operational complexity that requires highly specialized knowledge, not to mention the labor-intensive sample prep we talked about earlier.
But, despite those challenges, the ultimate verdict from the scientific community is loud and clear. As the source material states, mass spectrometry significantly enhances scientific knowledge and product development. It really is a foundational pillar that supports almost every single global industry and scientific discipline operating today.
And the craziest part, the journey doesn't stop here. The tech is constantly evolving, which leaves us with this provocative thought. As emerging technologies integrate artificial intelligence to rapidly interpret these highly complex mass spectrums, and as the massive hardware we looked at today gets miniaturized into truly portable devices, how is mass spectrometry going to transform remote fieldwork and routine, everyday healthcare in the near future? I mean, imagine a world where this immense analytical power literally fits inside a doctor's pocket or a researcher's backpack. The invisible world is only going to get clearer. Thanks for joining me on this explainer, and keep questioning what you can't see.